All right, so now that you understand a little bit about the components of cells, we're going to take a look at the cell's life, specifically when cells divide and when they die. And there's a few different things we need to discuss as far as that goes. So to start with, we have a process called mitosis. Mitosis is cellular division, normal somatic cells forming two new cells that are called daughter cells from one original cell. Okay, somatic cells only, not in gametes. Gametes are cells that become sperm and egg. Okay, these are the other cells of your body. New cells are called daughter cells. And then we have apoptosis, and apoptosis is programmed cell death. Cells need to die, otherwise development can't occur. Okay? So a cell lives in something called a cell cycle, and the cell cycle describes whether a cell is dividing or not. Okay? Different types of cells have different rates of cell division. So intestinal wall cells, the lining of your intestinal walls, those cells divide constantly throughout their lives. But brain cells don't divide. We already know sort of that, once brain damage occurs, it doesn't repair itself. Okay? Mitosis is important for growth. Growth doesn't occur without mitosis, and that includes like healing growth. The idea of mitosis or cell division is a very small part of the entire cell's lifespan. And you'll see sort of that it's this little part at the top, but most of the time the cell isn't dividing. It's actually doing other things. This phase called G1 or S phase or G2. And we'll take a look at what's involved in all of those. Okay, so the stages of the cell cycle are mostly interphase. So a cell lives in interphase most of the time. G1 and G2 phase, those are called the gap phase, and the S phase or the synthesis phase. There is a G0 phase, but not all cells go through it. It's sort of an optional, like a timeout kind of. And then we have mitosis. And mitosis has these common pieces that you should be familiar with. So prophase, metaphase, anaphase, telophase, and cytokinesis. Okay? We're going to take a look at interphase since that's the phase during which most of the cell activity or most of the cell life occurs. It's the most active time for cells. The normal things that those cells do, whether it's make proteins or contract and relax as in your heart cells or conduct nerve impulses, all of those things happen in interphase. Towards the end of interphase, we will replicate the DNA as the cell gets ready for dividing. Okay. There are two gap phases in interphase, G1 and G2, and then one synthesis phase. Occasionally there's a timeout phase called a G0 phase. Okay. Cells in interphase, this is a plant cell in interphase, not really interesting to look at as far as the nucleus goes. The genetic material, the chromatin is spread out throughout the entire nucleus. You can't see any chromosomes. But most cells that you look at will look like this because they'll be in interphase, because it's the most time happens in interphase. The first gap phase, or G1 phase, occurs right after a cell is finished dividing. So right after it divides, it goes into G1. And the job of the cell at that point is to build up its concentrations of molecules that it needs to do its normal job. Okay? So it's going to be making proteins and it is going to be making lipids and carbohydrates and all of those things. And they sort of surround the new daughter cells. From cell type to cell type, this phase, gap 1 phase, is the most variant. Some cells have a very short gap phase, some have very long gap phases, and all different phases in between, because depending on what the cell does normally, it may need to make a lot of stuff, or it might not need to make very much stuff at all. Skin cells don't make very much in the way of proteins. They make some keratin, elastin, and melanin, and that's about the extent of it. So their gap phase is pretty short. But cells like liver cells or intestinal wall cells, they need to make lots and lots of stuff, so their gap phase is fairly long. Okay? So anything that's slowly dividing will have a long gap phase. The synthesis phase, or S phase, this is where the cell copies its DNA. Okay? Now normally, remember, you have two copies of your DNA in the nucleus of every somatic cell. So when you copy that DNA, you now have two copies of two copies, which is four copies. And so each chromosome, each copy of the same chromosome now has another copy as well. and those pairs are joined at something called the centromere. And they look like little X's. So sometimes you might see pictures of chromosomes that look like X's. Well, that's actually two copies of the exact same chromosome joined at the centromere. In your body, the S phase takes about 8 to 10 hours. It takes a while to copy all of that DNA. Okay? During the S phase, we make the necessary machinery to separate the DNA into two sides of the growing cell that's about to divide, and the machinery to cut the cell into two pieces. So spindle proteins and centrioles are also going to form. G2 phase, after the DNA has been replicated, the G2 phase starts. And during the G2 phase, the cell needs to build up some energy and molecules that are necessary for cell division. Now, since we're making two cells from one, and we don't want those two cells to be very, very, very small, we have to make a lot of new membrane so that the daughter cells will be about the same size as the original cell that they came from. So we have to make a lot of new membrane during this phase. Okay, that membrane is made in the smooth endoplasmic reticulum. Remember that from our cell components videos. And it's sort of stored as empty vesicles, and those empty vesicles will eventually join with the larger membrane to make the dividing cell big enough so that the daughter cells are the right size. So once we're done with the second gap phase, we enter mitosis. Okay, and during mitosis, you can see the chromatids. Those are the chromosomes. Okay, as long as you stain them. And a chromatid is both copies of the chromosome joined at the centromere. They look like X's. Okay, during mitosis, those two copies are pulled apart, and one copy goes to each side of the cell, so that the new daughter cells each have a copy of that. You may remember the stages of mitosis. We just talked about them a second ago. We're going to look through them. Now, cytokinesis, the last stage over here, is technically not necessarily considered part of mitosis, but it is the final step, and it's important because that's the point at which the cells are going to finally separate into two new cells. Okay? Prophase. DNA coils up, shortens up, thickens up. The chromosomes thicken and kind of become visible. The microtubules assemble and form spindles, which are going to be attached to the chromosomes and help pull them apart. And the nuclear membrane, this is important, the nuclear membrane actually breaks down. So there's no longer a very defined nucleus. And that's important because we want to separate the DNA, and so we've got to get the membrane out of the way. So this is an animal cell in the stage called prophase, and you'll start to see the darker lines at the center are where the chromatids are being shortened and thickened so they can be seen. In metaphase, the chromosomes attach to the spindles at the centromere so that they'll be able to be pulled apart, and they align along the equator of the cell under great tension. So this is, you can see that this black line, sort of messy line at the center, that's all of the chromosomes lined up at the center. And the little faint lines that come away from those are your spindles. And those are there pulling on the two copies of each chromosome, getting ready to rip them apart from each other and pull them to opposite ends of the cell. Notice, no more nucleus anymore, and the cell is starting to be a little bit football-shaped. In anaphase, we start to see an indentation at the equator of the cell. There's a band of microfilaments, sort of like a drawstring on a drawstring bag, that starts to pull this thing closed to separate it into two cells. The centromeres break apart, and the spindles pull a copy of each chromosome to either end of the elongated cell. So that's what this looks like. You can see the chromosomes are being pulled to either side, and you can look, if you look carefully along the plasma membrane, you'll see the indentation about at the equator. That's where the drawstring is starting to pull it closed. In telophase, we get a dumbbell-shaped cell, chromosomes at each end. The spindle falls apart because it's done its job. It's moved the chromosomes to the sides, and the nuclei start to reform. The nuclear membrane starts to reform. Chromatin becomes more dense, and you can't really see the chromosomes anymore. So here our cells almost completely separated. We've got two new bunches of DNA on either side, and you'll notice the cell's pretty big. The daughter cells are going to be pretty decent size because of all that extra membrane. The last step is cytokinesis. The organelles and macromolecules are redistributed between the two daughter cells as evenly as possible. This microfilament band, this drawstring, contracts completely, which separates the two cells into two distinct cells. and the nuclear membrane reforms completely. In plants, it's very similar. Plants often have fewer chromosomes, so they're a little bit easier to see. So here on the left is interphase. Can't really see the chromosomes, but on the right, in prophase, you can start to see them. They look like little worms in the center. The nuclear membrane is starting to disappear. There's metaphase. They're lined up along the equator of the cell, and anaphase, they're being pulled, and you can kind of faintly see where the membrane is starting to indent. But in plant cells, they also have this cell wall, which makes it a little bit more difficult to see that. And finally, telophase, we start to see the plate in between the cells forming, and we can no longer really see the chromosomes. So that's the extent of mitosis. It's pretty straightforward as long as you understand what each of the phases does. Now, in the next lecture, we'll figure out, how does a cell control that process? There's got to be some kind of control to it, but we'll take a look at that next.